Okay, class, let's talk about photoreceptors in the eye, give a discussion of that. So, in the eye, in the retina, we have rods and cones. These are the photoreceptors. What they do is to absorb light. They absorb light. Strangely, though, they're positioned in reverse to what I would think, because when you go back and look at the eye, the light has to come through this the through the anterior portion of the eye, shall I say anterior portion of the retina, and then pass through all of these layers that you see in the previous video, and then finally get to the photoreceptor, and it's really the tips of the photoreceptors where light is absorbed. Okay, so the receptive regions are called the outer segments. This is in the pigmented area of the retina, pigmented layer of the retina there, and they contain these visual pigments which absorb light. Okay, so in this drawing here, you can see, we'll see right here, that this is the area which receives the light. That's a cone shape, that's a cone, that's a rod. That's that outer segment. This is the pigmented layer of the retina. Melanin granules are here. This pigmented layer also has cells that can do phagocytosis. Then we come to this inner segment right here, which is in the neural layer of the retina. And then finally we come here to the cell body, rod, cell body, cone. And then this is synapses here. This is the synaptic on the bipolar cells. So go back to the previous video to see the lineup. Okay, so we go now to the photoreceptor cells. They're vulnerable to damage, but they have a quick turnover. They degenerate if the retina is detached. They have to be near this area because remember, the choroid area is the vascular layer, and so they need to have nourishment there. Okay, they're destroyed by intense light, and the outer segment here is renewed every 24 hours, so they do have a good turnover. Okay, as we said, these areas are phagocytized by cells within the pigmented layer of the retina. The functional characteristics of the rods, we're talking about the rods, which would be these. They're very sensitive to light, best suited for night vision. One photon of light can activate them, contain a single pigment, okay, and perceived input in gray tones. Pathways converge, fungi. So the pathways converge aspect means this, that several bipolar cell synapse on a ganglion, so they come together. So when you're looking using the rods, you can't get pinpoint sharp images. The cones, however, go one-on-one in to bipolar and then the ganglion cells. So there's a sharp image there. Okay. So we go further. Here's a comparison of the rods and cones. The rods are non-color vision. Okay. They only have one type of pigment, high sensitivity, function in dim light, very low acuity, sharpness of vision because of how they go into a convergent pattern, more numerous, and mostly in the peripheral retina. The cones are in that area, the macula lutea and the fovea centralis. Color vision there, low sensitivity. They don't pick up light easily. So in other words, their problem is you have to have more of a brighter light, but they do give visual acuity if you have the brighter light. High acuity, one cone per ganglion in fovea centralis, less numerous, mostly in the central retina. Okay. The visual pigment is composed of two things. a vitamin A substance called retinal hooked to a proteinaceous substance called an opsin. Now, all the retinals are the same. Retinal is the same. It's a vitamin A thing. It's the opsin that makes the difference. And there are 4 opsins. There's one for red, green, blue, and then the one that's present, and those in the cones, and then the one present in the rods. It's synthesized from vitamin A. So let's look at that. Let's go here. Let's go here. We'll come back. So here's vitamin A, known as retinol. Through enzymatic reaction, here would be retinal. Retinol is vitamin A. Here's retinal, which is the pigment substance, the retinal here. Okay, and we'll come back to that. So it's derived from vitamin A. Okay, there are 2 types that we'll get into, the 11-cis retinal and the all trans, and we'll talk about that in just a second here. So I go back here. Okay, I want to go back here. And we're looking at the one that's present in the rods that we call rhodopsin. Rhodopsin is an opsin, which is a protein, combined to the retinal, the vitamin derivative, vitamin A derivative. And so we're looking at the 3 steps of rhodopsin formation and breakdown. So that's where we are now. Okay, we're going back to the vitamin A again to look at retinol versus retinal. So the retinol, vitamin A, retinal conversion. Now, when I go to the retinal, I want to draw your attention to this here. There are 2 forms of this retinal, an 11-cis retinal and 11-trans. Let's look at the chemistry a little bit. Okay, here's a cyclic. Here, this particular right here has 15 carbons, you see right there. Okay, 1, 2, 3, 4. This is by nomenclature of numbering the carbons. Here is the 11th right here, carbon. Okay, a cis, go back to my chemistry, biochemistry in my ANP1, a cis has both side attachments on the same side. Trans has it opposite. So there are 2 forms of retinal, an 11-cis, 11th carbon, and an 11-trans. See, all the others were trans. This was the only cis. Because it was a cis, it bent the molecule. It bent the molecule. So I want you to keep this in mind as we look at this here. All right. So the pigment in the rod is rhodopsin. Again, 11-cis retinal initially with an opsin known as rhodopsin. That's the pigment. We're looking at the steps of rhodopsin formation. Okay, so what will happen is in the cis form derived from vitamin A, so the first thing you do is enzymatically derive it from vitamin A. When it's enzymatically derived from vitamin A, it's going to be in the cis form. Okay, all right. So enzymatically derived from vitamin A. Okay, once it's in the cis form, okay, and here's vitamin A converting to this 11-cis, which is a process of oxidation. Oxidation is removal of electrons, but hydrogens can go too, hydrogen protons, hydrogen ions. When you do that, then the 11-cis retinal can combine with the opsin, the protein. So the 11-cis then can combine. When light hits this in the rod, the light absorption by the rhodopsin causes the retinal to go from an 11-cis to an all-trans. When that occurs, it cannot attach to the opsin. So it detaches from the opsin. So now I have opsin separate from the all-trans retinal. And this is going to be present in the membranes, in the disc of the outer segment of the rod. The disc of the outer segment of the rod. Okay? Now, what will then happen is that enzymatic conversion will eventually take it back to become 11-cis-retinal. And what that activity involves is an enzyme that will cause that to occur. Okay? And also the use of ATP. So it's really a rhodopsin kinase enzyme. A kinase enzyme phosphorylates a protein, and that rhodopsin kinase enzyme, using ATP, taking the P away, will then come phosphorylate it and bring it back to this. So in other words, we're adding the electrons back. That's a reduction process. So it's going to oscillate between the retinal, 11-cis-retinal, and the all-trans retinal that occurs. And the process of removing the retinal from the opsin is called pigment bleaching. And this is one of the first steps in this action of phototransduction, pigment bleaching. So you can look at that. So pigment synthesis, pigment bleaching, and pigment regeneration coming back again, as we alluded to. Okay. I wanted to go back one more time to the disc that are present in the rod. What happens in the rod, unlike the cone, is that you have 4 membrane containing disc, surrounding disc, and here's the membrane of the cell itself. Now let's go here if I can pull this up. So here they're disc independently, like stacks of coins in here with a coin wrapper around them. That's the cell membrane. The disc in the membrane of the disc are where the photoreceptors. In the cones, you just have an enfolding of the cell membrane, but you still have your visual pigment within there. But we're only doing the rod. The cone is very similar as to how it does its bleaching and everything. So this first video, I'm going to say, is Phototransduction 1, the capturing of light. We're going to close this video out and start another one to take it further into an understanding of this process.